purpose, now we are looking at it in a different aspect. We are going to look at activity that happens within the mouth. We produce saliva, and saliva usually has some enzyme, and major enzyme being kaolin, or if some people choose to use salivary amylase. We do have some lingual lipase, but it is minimal amount. In humans, it's even negligible. So the main enzyme we'll be targeting here is the salivary amylase. As you go down, we do not have any enzymes within the esophagus, because the esophagus just acts like a transit or conduit to connect the mouth to the stomach. However, when we go to the stomach, this is another organ that produces enzyme, and the major enzyme being pepsin, although we also find some renin, but the enzyme that we'll be working on here is pepsin. Now, if you look at both saliva or rather salivary amylase and pepsin, they require different environmental conditions. This is slightly basic. It needs a pH that is slightly basic. So it means that if you are to alter the pH and you make it acidic, the salivary amylase will be rendered inactive. On the other hand, the pepsin working in the stomach requires an acidic environment, usually at a pH of 1 to 3. If you increase the pH beyond that, you find that the pepsin becomes inactive. So one of the experiments that you can do is to alter the pH of this enzyme and to see what happens as you keep on increasing the pH. You look at the rate at which the enzyme catalyzes the reaction. Similarly, you can do the same experiment with with the salivary amylase, but in our case, we won't be looking at the rates of reaction, but we'll be looking at if you have a different pH. So, for instance, if you have a basic pH versus an acidic environment, whether the reaction will be the same. The other thing is when we are dealing with enzymes, you also have to watch the temperature. So when we have very low temperatures, what happens to the enzyme activity? What will happen if you give it an optimum or close to the body temperature? And then remember that these enzymes are proteins. That means if you increase the temperature beyond a certain level, you're going to denature the proteins. When you denature the proteins, you render the enzymes inactive. So you stop the reaction when you increase the temperature. So by simply boiling the contents of a reaction, you find that you're going to stop the reaction because you have denatured the proteins. Now, as you go down the digestive tract, you're going to come to the duodenum. From my lectures, I told you that within the duodenum, we have the entry point whereby the contents of the gallbladder and the contents of the pancreas are released into the duodenum. So we have to check what is secreted within the liver stored in the gallbladder. This is usually bile. And what does bile target? It usually targets the lipids. So for now, we do not have an experiment set for you to target the biles. However, we have the pancreas here. The pancreas secretes pancreatic enzymes. Among the pancreatic enzymes, we will have those that will target carbohydrates, we have those that target the proteins, we have those that will target the lipids. So we are talking of things like trypsin, chymotrypsin, We have the peptidases. You can have the carboxypeptidases, aminopeptidases. We also have salivary amylase that will target the carbohydrates. We do have pancreatic lipase that will target the lipids. Now, all these enzymes are secreted within the secretions of the pancreas that we term as the pancreatic juice. So, this will be released upon stimulation, and it is released in the duodenum. So, as you can see, enzymes of the pancreas are going to target all sorts of food. It will target the proteins, it will target the carbohydrates, and it will also target the lipids. However, again, the environment in which these enzymes will work will be different from the environment of the stomach. So within the stomach, we had pepsin, and the optimum pH for pepsin is the 1 to 2, which is very acidic. This acidic mixture, once it comes to the duodenum, is usually not conducive or optimum for enzyme activity for the enzymes from the pancreas. So these enzymes require a basic pH. So how do they ensure that the environment here is optimum for the best enzyme activity here? So you find the pancreas, in addition to secreting these enzymes, is going to secrete bicarbonates. Bicarbonates are going to raise the pH within the duodenum. So as you release the pancreatic juice, you are also releasing a very basic or alkaline media. And this alkaline media will neutralize the acidic component coming from the stomach. Now, in addition, the walls of the duodenum are going to secrete also bicarbonates. So this combined effect of the walls of the duodenum and the pancreatic juice, and in addition, even the bile component will have, or rather, even the components from the gallbladder will also have bicarbonates. So we have a lot of bicarbonates being secreted here, which will neutralize the acidic pH coming from the stomach, and at the same time, they will raise the pH, making it optimum for these enzymes to work. Now, the enzymes will act on the food, and it will break down. If it is the lipases, they will target the lipids. If it is the carboxypeptidases, aminopeptidases, they are going to target the proteins. If it is the trypsin, chymotrypsin will also target the proteins. If we have the pancreatic amylase, this one will target the carbohydrates. So you have all those foods being broken down, and they'll be broken down usually to dimers. That means instead of having a single glucose joined to glucose, you can have glucose to glucose, you can have glucose to fructose. So you find that you will have simpler molecules, but they may not be broken to the individual building blocks. So what you're going to find is that within the walls of the intestines, we also have some enzymes that are going to completely break these bonds and release the individual monomers. And these monomers are the ones which will then cross the wall and get absorbed into the circulation. Now, what I need you to note is that I'm focusing on where the enzymes are coming and the environment in which they work and the substrate that they target. And finally, when they target that substrate, what product will they get and where does it go? So far, we had carbohydrates targeted in the mouth, we have proteins targeted in the stomach, and then we have all types of food that are targeted within the duodenum and the small intestine. Once they are broken down to the building units, then they can cross over for absorption. However, not all these foods will be absorbed. Secondly, these enzymes are also proteins. So as they are released, you'll find that some of them are actually going to be broken down and they can also be recycled or reabsorbed. However, you find that not all these enzymes, as they are released, will be broken down. So some of them actually travel the rest of the GIT together with the waste products that will be eliminated. And you find that at the point of defecation, we'll also have some enzyme activity in the fecal matter that is released. So in order to check for enzyme activity, you can actually take a sample of the fecal matter and address it and actually process it so that you can see if there is any enzyme activity. So one of the activities that you're going to do in the lab is actually using fecal matter so that you can look for enzyme activity. And the main enzyme that we target is trypsin, because trypsin is the one that is secreted in the highest amount within the pancreas. So if you want to check for pancreatic activity, you want to see if your pancreas is producing trypsin as it should. Then the trypsin, once released in the duodenum, will traverse the rest of the GIT and it should be released in the fecal matter, and you'll be able to work with the fecal matter to look for trypsin activity. So I think the next slide is talking about the amount of secretory activity that we have. We have a lot of secretion that is released throughout the digestive system, but I have talked about most of the components, which are mainly the digestive enzymes, although we also have mucus, which will allow lubrication. We also have bile, we also have electrolytes. These ones I mentioned when we were in class. So the main glands that you target are the salivary glands, because they're the ones which are producing saliva. We have pancreas because it's producing the pancreatic juice, but we also have liver that is producing the bile. So these are usually from the accessory organs, but we also have the walls of the GIT also having some secretory activity. So if we are to focus now on enzymes, because I have mentioned the types of enzymes and we have mentioned where they are coming from. Enzymes of the digestive tract, just like any other enzymes, are proteins. So you produce them as you normally produce proteins, but then these proteins are specific for the substrate that they catalyze. I'm sure you're going to do a lot of these in biochemistry, but I will focus on the ones that we'll be working on in the lab. So the enzymes are generally biological catalysts in that they will enhance or they will affect the reaction, but they themselves, at the end of the reaction, they are not affected. And then they are able to catalyze other substrates. So you just require a little amount of enzyme and to keep on catalyzing the same substrate, and itself, it is really not affected. Most of the reactions of the digestive enzymes are hydrolytic activity. That means they will add a water molecule to split long molecules. Usually, storage material or food that is stored will take the basic monomers, and as you catalyze the links between the basic monomers, you usually release water. Now, in the case of digestive enzymes, you'll take a water molecule and use it to split them into monomers. So that's why we say they have hydrolytic enzyme activity, or they are also called hydrolysis. Now, you find, like I mentioned before, every enzyme is very specific for the type of substrate that it works on. So, for instance, I have just mentioned that we have amylase. Amylase can be from the salivary amylase or the pancreatic amylase. However, the substrate is the same. It will be targeting carbohydrates. If we have pepsin, we have trypsin, it will target only proteins. Now, since you find that the digestive enzymes are usually acting outside the cell, so they are being released by epithelial or GIT epithelial cells, or they are being released by the glands and they are released into the lumen. That means their reactions or activities are done outside the cell. For that reason, you can actually harness that and utilize it to your advantage in the lab. You can check the hydrolytic activity within a test tube of whatever you are doing. And you find that this is what we'll be doing during the practicals. For us to understand what it is happening, we'll be looking at digestion of the different foods. I may not focus so much on the absorption. I'll do that when we're doing the theory. But for now, I want us to focus on digestion. For you to understand, you need to know which are the different classes of food that we have. The major ones being the carbohydrates and the proteins and then the lipids. So if we look at the carbohydrates, the ones that we usually ingest are mainly the starch or the disaccharides. So when we talk of glucose, like when we take, not glucose, but sugar, when you talk of sugar, you find that it is made up of sucrose, and when you break the sucrose, they are actually disaccharides. Alternatively, you have starch that is stored in plant material. Or if you are to take some liver, you find that it has a lot of glycogen. So the question is, these carbohydrate monomers, what are they made up of? So they are simply made up of simple sugars, which could either be fructose, glucose, galactose. Now, when they have a combination, they combine to form the disaccharides. And when you have a link between two monomers, you form what we call the glycolytic bond. So when you have to split to the individual monomers, you have to have an enzyme that is able to cleave or to cut that bond. So when we have starch, starch is organized as chains of sugar molecules, mainly glucose. And this glucose can either be straight or branched. And once it is branched, it will form one type of storage molecule. So I will capture that in class so that I can show you the different molecules that you can come up with the long chains of glucose molecules. What I want you to note is that in all these carbohydrates, we have that glycosidic bond, and it is the one that will be split. What you require to split that is usually the amylase. Now, if I were to draw here, I usually find it difficult to teach without drawing. So if we have the glucose molecules here, so I have the glucose linked to a glucose. This one gives me a hard time. So I have, let me just forget about drawing. It is not allowing me to. So when you have a link of glucose to glucose to glucose, and you take something like a big chopper, a chopping knife, and you just chop once, you're going to have two chains of the glucose, or rather of the starch molecule. Chop again, and you'll have shorter chains. Keep chopping, and what you'll be doing is you'll be cutting those long chains of the storage molecule into smaller chains. So the work of the amylase is to act like a chopper, like a chopping knife. It will be cutting these long chains into smaller chains of these glycolytic or saccharide molecules or polysaccharides. Now, once you chop them, this will happen in the mouth because the first place they are going to encounter is amylase within the mouth. So this amylase will be chopping it into shorter chains or carbohydrate chains. These are the ones which will be taken into the esophagus. Once they go into the esophagus, they will get into the stomach. I want you to keep note that in the stomach, the environment is too acidic for this salivary amylase to work in. So you find most of the carbohydrate digestion will be halted in the stomach. However, these shorter chains that were released, once they are taken into the duodenum, they will be exposed to pancreatic amylase. So the amylase will continue the work of digestion that was started by the salivary amylase, and this occurs within the duodenum. Now it will chop those longer chains. Remember, we had chopped them into shorter chains of carbohydrate chains. Now, when you chop them, you'll form short chains of which we'll call the oligosaccharides, or dimers of glucose of simple sugars, which we are going to call the disaccharides. So they could be maybe chopped to 2 monomers, dimers, tetramers, just short chains of these molecules. This, remember, all this is happening in the duodenum, but the duodenum is continuous with the jejunum. And so what will happen is that as they go to the jejunum, to the ileum, they will come in contact with the epithelial cells of the walls of the intestine. Remember, I told you about churning and mixing. So as you chop them, they are thrown towards the walls of the GIT, and when they go there, they will come to the epithelial lining, and within the epithelial lining is embedded some enzymes. Remember, enzymes are proteins. So these walls of the GIT, the epithelial cells, exposed to the lumen side, we have what we call a brush border. This microscopic extension from the cell membrane. Embedded within them are enzymes that are known as the brush border enzymes. These brush border enzymes are the ones which do the final chopping of these oligosaccharides and the monosaccharides, so that it chops them and releases the simple sugars. The simple sugars being the glucose, fructose, or galactose. And now, in this format, you are ready to absorb the food. So now, if you already know, we have started with the carbohydrate in form of starch. Or if you took some animal product, then you have the glycogen. You've chopped them down, and what you end up with are the simple sugars. So if we were to do an experiment in the lab, we can look at the activity of amylase. And because between the salivary amylase and the pancreatic amylase, the one that is easier to access is the salivary amylase. So in the lab, you are going to produce saliva, and you are going to use that salivary amylase. You are going to expose it to a starch solution, and given different conditions, whether it is temperature, pH, and the amount of enzymes, or you denature the enzyme, you are going to expose a substrate to the salivary amylase. You are going to test for starch before the experiment or after the experiment. Remember, you don't really have to test before and after. What you can do is to produce several tubes, one in which you are going to put the salivary amylase. So that shows you the contents before digestion. Then one that you put salivary amylase where you can alter the pH, and another one where you can change the temperature, and another one where you can actually give two optimum conditions and you remove the other one. But the final one, you have to have a control whereby you know for sure you have a starch which will be digested by salivary amylase, or if you have an enzyme that you are given to act as a positive control. So you normally have in the lab a negative control and a positive control to show what happens or what color or what changes you expect when the activity does not take place as expected. And a positive control is when you give all the optimum conditions so that you expect positive results. So in this case, because we expect amylase to act on starch at an optimum pH of, say, a neutral pH, and probably the best temperature will be around the body temperature. So if you give those conditions, you are likely to enhance the activity of the amylase and you are likely to get positive results. Which means if I take this tube where I have given all the optimum conditions and I allow it some time, then I test for starch. You find because amylase will have broken down the starch, I'm likely to get a negative result for starch. On the other hand, what are the products of the amylase after you digest them? The products are likely to be these simple sugars, which means at the end of the activity, if I test for simple sugars, I'm likely going to get a positive result for sugars in this tube where I had given the optimum conditions. So my digestion will have occurred and I will have presence of the simple sugars. So far, any question regarding that so that you can see what you're going to do in the lab? Any questions? I don't see any hand up, so I will proceed. So, one of the ways to test if digestion has taken place is to test for simple sugars. You can also test for starch. So there is some you'll be given some of the materials that are used to test for starch, and you'll be given what is required to test for simple sugars, and you look at the end result. So after your experiment, which tube is showing the presence of digestion? So that will be your first experiment. Now, if you notice, I try to avoid telling you take tube A and add this component, because that is technical issues, which I expect that you will discuss with your technologist. He's the one who will tell you which tubes, how many tubes you should have, and what to put in each individual tube. So I'm not going through the technical component of the actual practical. I am explaining to you the theory behind it. So for those people who are not taking notes, remember that these pre-lab sessions are the ones that you're going to utilize in your discussions. so that you explain the results that you observe. So now, having done the carbohydrate digestion, the second enzyme will be addressing protein digestion. So before we do the protein digestion, we need to see where do we digest our proteins. So we normally have, although you start breaking down mechanically the proteins in your mouth, you chew them into smaller portions. However, there is no enzyme that can digest the proteins in the mouth. So after chewing, this foodstuff will be swallowed and it will settle in the stomach. Now, in the stomach, it will find what we call the gastric or stomach juice. The stomach juice or gastric juice is produced by the stomach glands or the glands within the walls of the stomach. One of the things that it is rich in producing is hydrochloric acid. So it will produce a lot of hydrochloric acid, which will create an optimum pH for the pepsin, which is also an enzyme produced by the stomach lining. So the pepsin will be produced and it will act on the proteins. It is also important to note at this point that pepsin is not produced as pepsin as say, it is produced as an inactive protein. Remember, even this is an enzyme. And this enzyme, if it is active, it will start digesting the walls of the stomach because they are also made up of protein. So to prevent that from happening, pepsin is synthesized in the form of an inactive protein known as pepsinogen. This pepsinogen is released, but it will not become active until it is exposed to hydrochloric acid. The hydrochloric acid will aid in splitting or cleaving one portion of the pepsinogen to release the active pepsin. Now, the active pepsin will target the proteins and it will start chopping them down in a similar manner to the way amylase was acting on the long carbohydrate chains. So now we have large proteins, and these large proteins, they are highly folded. Remember, a protein is made up of amino acids. Amino acids folded and folded into secondary and tertiary structures or even quaternary structures. All these are held in place by hydrogen bonds. So they are held together by the sulfide bonds or by hydrogen bonds, which keep the proteins in shape. Now, when you come and chop, some of these weak bonds are still holding the structures together, but the presence of the HCl will help in breaking these small hydrogen bonds or disulfide bonds, making the proteins linear. When they are made linear, they become accessible for other enzymes such as the pepsin to start chopping them into smaller peptides. So the end products of what is released from the stomach are actually polypeptide chains, just chains of amino acid linked to amino acid linked to amino acid, forming short peptides. These are the ones which will be passed into the duodenum. Now, this will be acted on by the pancreatic enzyme. So you remember, I told you within the duodenum, we have enzymes that are released from the pancreas. Among these enzymes are the trypsin, chymotrypsin, the carboxypeptidases. They are going to start cleaving the bonds that hold the amino acids together. So these bonds are usually the the peptide bonds. And what they are going to form are the polypeptide chains. So you are going to use pepsin or trypsin or chymotrypsin or the peptidases to cleave those peptide bonds. What you'll end up with are either single amino acids or amino acids joined to amino acids to form the dipeptides, or these dipeptides having an additional amino acid to form the tripeptide. So there will be very, very short chains of peptide molecules. This again, like I said, all this is happening within the duodenum, and as they move down to the ileum, now you have all these dipeptides, tripeptides, or single amino acids. These products will be exposed to the brush border enzymes, and the brush border enzymes will cleave the final peptide bonds to release the individual amino acids. And now this will be ready to be absorbed into the circulation. So now you know that most of the protein digestion will happen either in the stomach or in the small intestines. So we have the enzyme pepsin, which is working within the stomach, and it is acting in an acidic media. We can utilize that knowledge to set up an experiment to see how are proteins digested. And what we'll do is you can use a source of protein, something like egg white, which has a lot of protein, or you can use ground beef, or you can even use beans, any source of protein. When you provide the optimum pH, optimum temperature, and you expose them to pepsin, then you find that these proteins will be broken down to the short peptides. So we can do this in the lab, where you'll be provided with pepsin and HCl, and we'll check the activity of these enzymes. So what you'll find is that you test for proteins, and then you see what happens after digestion. Now, I told you that there is another experiment which also targets the protein digesting enzyme, mainly trypsin. In this case, you are expected to use a stool, or you are expected to use another protein source. Now, you find that you can have a matrix. You can make a matrix made up of jello or jelly. When you take crystals of jello and you produce, and you actually boil in water so that it forms a protein matrix, if you expose that matrix to trypsin or to even pepsin, what it will do, it will digest the protein that is in that jello. Now, when it does that, we find that it will not solidify. So another experiment that you can use, even if you don't use the fecal matter, you can use jelly crystals to produce a protein matrix, but in the presence of trypsin, you're going to see if you are able to produce such a matrix. Now, most of these enzymes we are talking about are produced. They are actually within animals, so they are released by animals. But that doesn't mean that all enzymes have an animal source or they are of animal origin. So we also have another experiment that will be targeting proteins, but in this case, this enzyme is from a plant product. So we are going to use papaya. Just give me 1 minute, please. Okay, how many are still with me? Or you ate ugali and slept?
We're still with you.
No.
Yeah.
All right. Yeah, I just want to know that I'm really not talking to myself. You know, I'm in the office, so I look very weird just talking to a computer. So now we will have we have some plant enzymes, and one of the I would say popular one is one from the plant pawpaw. You know what the pawpaw fruit? It produces an enzyme known as papain, and we will be extracting some papain from the pawpaw plant, and we will be utilizing it also to look for protein digestive activity. Again, I will not dwell into the technical component or the technical aspect of how you do it, because if I do that, then your technologists will have nothing to tell you. So I need you to also interact with them so that you can understand why you're doing the practical the way you're doing it. So all that will be targeting trypsin, pepsin, and papain. Now, the other experiment that we utilize is using an X-ray film, and this one targets the trypsin found within the fecal matter. So you'll take some fecal matter and you're going to process it by adding, just diluting it so that you're not working with solid stuff. Once you dilute it, what you're going to do is clean it up, because you have some filter papers, you have all that, pour it up, and when you have all that clear fluid that you can work with, you're going to dip a strip of X-ray film and allow it to sit there for some time. And then after that, you're going to leave that X-ray film, and after rinsing, you look through and see what happens. Now, this is where I'll target most of my questions, because I want you to understand and to do the practical, that is using the X-ray film. If you don't do that, you will not understand. And this is where I will target most of my practical questions. So we have always done it. I have done it several times. Your peers ahead of you have done it. So I don't see what is the big deal about doing this experiment. Now, the next one will be targeting digestion of lipids. Now, with lipids, we can have minimal digestion by the lingual lipase, which will be found within the saliva, within the mouth. However, most of it will be swallowed. Most of the fatty foods that you have or lipid products will end up in the stomach. Within the stomach, again, there is minimal digestion within the stomach, and it will move all the way to the duodenum. Now, in the duodenum, it will be exposed to the pancreatic enzyme lipase. However, remember that these are fatty components. They do not dissolve in saliva, because saliva is mainly made up of water. When you release them into the stomach, we have the gastric juice, which is again mainly made up of water. So you have these chunks of fat, or if it is liquid oil, it is floating oil. And you find that when the lipase tries to get to those individual molecules that make the lipid, it has a challenge. For that reason, you have to break up those big chunks of fat or those big chunks of oil into small, small particles or droplets that can be worked on. To do this, we need bile salts, which act like a detergent. What does a detergent do? I don't know whether this will allow me to draw, but When we have a big chunk of fat like that, and you slash it physically to get two smaller chunks, or even oil, if you have a very large droplet of oil and you split it physically, you get two droplets of oil. The problem is if you leave these two droplets to come together, they will reform that big chunk. If they are fat droplets, you have fat chunks, you find that when they come together, they will stick to each other. Now, if you add a detergent, what a detergent will do, it will come and sit on the surface of this chunk. And then when you split it into two, it will sit on the surface of all these. And what will happen is because of this presence of the detergent on the surface, once the droplets have been cut into two, they cannot reunite. Again, if you shake this and you end up having again smaller droplets. So now you have these smaller droplets, whether it is oil or fat chunks, now they are smaller. Again, because of that detergent, remember it is sitting on the surface. Again, you end up having this detergent coming here. So what in essence you are doing is you are increasing the surface area of these large droplets, because finally you are going to have very, very tiny. You have very, very tiny droplets. And these droplets cannot coalesce together. So when you have those droplets, I hope I'm seeing that. Okay. So when you have these tiny, tiny droplets, they do not coalesce together, they do not reunite. But what you have really done is that in essence, you really increased the surface area of the fat. When you increase, it means now the lipase can come and access the tiny molecules. So the lipase will come in between all these droplets and it will start cleaving the lipids to release either glycerol molecules or diacyl glycerols or monoacyl glycerols. So now when you have those, they are ready to be taken in by the process of absorption. So for fats, the major thing that you can do in the lab is to observe emulsification process, which will be the splitting of these large droplets into tiny droplets by the addition of a detergent. This is the same process that happens in your GIT. So what is acting as a detergent? The detergent is coming in form of the bile salts. So we have bile, which is producing bile salts and bile acids. These will come and they are released into the duodenum. And you find one of the characteristics of the bile components is that they have both a hydrophobic part and a hydrophilic part. So they are amphipathic in that they have two portions, one that is lipid loving and one that is lipid hating. So what will happen is that the lipid loving will come and sit into these fat areas and they will expose the water loving portion to the surface. That way, if the water loving portion is on the surface and these droplets comes in close vicinity with the other one, they cannot reunite. That's why the bile is acting like a detergent, in a similar way as when you add liquid detergent to an oily substance, even when you are cleaning dishes in your kitchen and they are all layered with oil, if you add detergent, it is going to emulsify or to break down those oily substances into very, very tiny droplets. So, a good experiment to show this emulsification process. If you take a bottle and you fill it halfway with water, and then you put a layer of oil, you find that the oil will sit on top of the water because the oil is usually lighter than water. If you keep the bottle and then you shake it vigorously, you find that although the oil will break into small droplets which will disperse in the water, the minute you put down the water, all those droplets will rise to the surface and they will reunite and form an oil layer. However, if you take that container and you add a drop of detergent and you repeat the process, you find that every time that oil droplet breaks, it cannot coalesce again. And when those ones drop, so if you shake vigorously and then you put that bottle to sit down, you find that the water becomes cloudy. And the cloudiness is because we have all these small microscopic size droplets of oil that are dispersed in the water. But because of the presence of the detergent, they cannot coalesce again and form that oil layer. So that is a simple experiment that you can do in the lab showing emulsification of oil. And it is the similar process that occurs within the GIT. So I think having done that, I will leave the second part of the pre-lab because as you can see, this is addressing abdominal sounds. The reason why it is done that, I think it will be done in the second lab where we'll be listening to abdominal sounds using a stethoscope. So when it comes to that, I will explain to you what happens within the chest and what happens within the abdomen. So I will skip this pre-lab to the second session where we will be addressing sounds that are coming from within the body. For today, I wanted us to focus on enzymes and to relate them to the practicals that you're going to do in the lab. So, any questions? I told you it's a short pre-lab lecture which should take 1 hour, and I think I'm right on time because I started at 1:35 and now it is 2:36. Any question? So there are those people who I know they did not pay attention. I did not do that participation so that I can include everybody. But what will happen is that if you are not listening to this pre-lab session, you'll have it rough when it comes to writing your reports. Yes, Cecilia.
Are we going to get the lab books or we are going to do as we did last semester?
Thank you for reminding me. I'll find out because I don't know whether the books were availed. It took a long time for us to get the books. So I'm hoping this time you have the books.
Another question is, we will use the same format in writing the reports?
Yes.
Thank you.
Okay. Any other question? So for those who are recording, you can stop recording.